Clementine, I see you will take the minutes. Clementine?
Hello, everybody.
Hello.
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 Okay. Thank you for joining so late, Clementine. I'm sure it is late night for you. All right. So according to the schedule, yeah, I'm chair and you, Sid, is minute taker. Okay. Is that the way we're going to do it, or are we still out of kilter?
Sorry, my audio just wasn't working, but I'm happy to take minutes.
Okay. Cool. Thank you. All right, we'll get started in a minute. Just a few intros for new people. So joining us on the call is Rosie Chen, who is interested in open science and how we work as a group. So I sort of suggested that perhaps you could come along to take a look at a meeting and see what's going on, just for information. Welcome, Rosie. And I guess, Sean, we haven't met before. Is that right?
I think that's correct, yes. Wendy asked me to if I'd be interested in coming along, so here I am. I'm based at Maynooth University in the Republic of Ireland, and we've done some work with Wendy over the last year or so. So I'm delighted to be along. Thank you very much for the
All right.
invite.
You're very welcome. So I guess we're talking about targets and other things later on in the meeting. I guess that's the point where we might come to you for some input, I guess. All right. So hopefully everyone has the agenda, sort of outline agenda for today, which was put on GitHub. I will just put that in the chat in case there's any doubt about it. And today we are hopefully going to spend some time talking again about new potential targets. But there are just a few other things we should go through swiftly if we can. If I could just jump to the Series 5 paper, because I spent some time on that last week and I've just taken the comments from Andrea and Charlotte on that manuscript, and I've incorporated everything, and we've got a sort of clean version, which I will share on GitHub right after this meeting. And there are just a few comments that we need help on from Wendy and from Louisa in that manuscript. So we don't have many edits left, but there are just some comments on the right. And if you wouldn't mind, after the meeting, when I've posted the new version, just going through those comments and providing anything that we can. That would be really great, because we're nearly ready to go. And essentially, there's nothing major left now, which is great. We just need those last few comments done. Okay. And then there was, so that was the update on the Series 5 paper. We can, if you want, Clementine, talk about Series 2 paper while we're talking about papers, if you like. Is there anything you'd like to bring up?
I don't mind what order we do it in, but I have a fair few things to bring up, so maybe it would be better to leave it to later.
Fine, let's do that. All right. If I forget, then just jump in. There were just quickly this in the last month, a couple of new issues that have been brought up or related to new results that have been generated. So let me, I might share screen just for a second because it would be useful to have a few comments about this. 1 second. Just in terms of it would be useful to know if we have to do anything So hopefully you can see my screen now. So there were two new areas of molecules that were evaluated. One is the on-code library, which Wendy posted. So there are some new structures that are featured there that have come out of that screen. These, to my naive eye, look unpleasant molecules that might do lots of different things. But I think there was some general consensus in the comments that these comments didn't seem to be generically toxic, and B, there may be some target that was being suggested by Bernard. Did anybody want to address these molecules? And I haven't followed up
Yeah.
steppers.
So those salicyl analyzers and niclosamide derivatives, I went looking on a site called FRAC, which I think is like Fungicidal Resistance Action Committee or something like that. And these salicylanilides, there's a compound that was used in the 70s called benodinil, which is salicylanilide with the hydroxyl replaced with an iodine. And it inhibits succinate dehydrogenase, or complex 2. So that's probably another possible target. And I don't know if there's any assays available for that, but it'd be interesting to see if niclosamide inhibits fungal succinate dehydrogenase.
Okay. Any other comments while we're on this page?
Yeah, I think we can do this together with the colleagues in York. I think they have already suggested to prepare some derivatives of the benzamide or salicylanilide type of compounds. And we have also asked Dr. Melander from Notre Dame University if he has some samples left he might send to Wendy, samples which are related to that IMD compound, benzamide or salicylamide derivative.
All right, so that's been budded off into this.
Yes, I just wanted to ask Wendy if Dr. Melander has already answered about his plan, which compounds he wants to send, because he wrote 2 weeks ago that he will write an answer last week, but I haven't received any email from him. Maybe Wendy got a reply from him about that. That's what I want to ask now.
No, I don't think we got a reply yet. We said how much we would need roughly, and that's it.
Okay, then I will contact him again and ask if he has any news about that quite interesting compounds.
Yeah, and what we concluded in the discussion of this paper was that these compounds most likely will have a dual activity, so against the host and against the pathogens. And since there's a large inflammation zone surrounding the mycetoma grain, that might not be necessarily a bad thing.
Yeah, the interesting thing is that his compounds, Dr. Melander's compounds, were developed to fight bacteria. So they should be also quite selective and not too harmful to the host organism, I suppose.
Okay, great. So the next step would be to try and secure samples and evaluate those to I guess see whether or not there is activity in this series.
What were agricultural fungicides already screened? Because there's, I believe, about 3 or 4 of that sort of benzamide type. So if they were screened.
Yeah, I put the list in the large compound list. It has its own tab. I haven't looked specifically at what were agricultural fungicides, but I marked the ones that were potent yellow.
So what Dunja meant is that she screened a library of fungicides, antifungals, a known library with known compounds. We didn't do anything yet with it further, but she posted it on the database as a separate tab. So,
Okay, yeah, that's great. I'll go check that right now, because there's this compound called boscalid, which is basically a salicyl analide, but it's got like some isosteric stuff going on, bioisosteric stuff going on. So I'll take a look at that right now. Thank you.
Yeah.
Okay, great. And then the one other small thing was that Clementine opened an issue for just some log D calculations, I think it is, which we were hoping, I think, that Louisa could help us with, if possible, if that's right.
Yeah, there was some special software that was used that was proprietary, I think.
Yeah. Yeah. So if that's a possibility, that would be very helpful. All right, so those are the sort of main things, apart from the discussion at the end that we can have about the Series 2 paper. So I guess we can then pivot to the main thing that I think we'd like to talk about today, which is trying to think about what might be the best new targets that we could go after as a more target-based approach rather than a phenotypic approach. So we're quite keen to try and think about better targets for future series. The action that was on me to reach out to the SSG CID folks, so the Seattle Structural Genomics SSGC Institute for Infectious Diseases, I think it is. So they do crystallography, mainly protein expression and crystallography for targets related to infectious disease in Seattle. So I have reached out to them and asked them if they happen to already have any proteins from Madurella that we could use. And if they don't, whether they'd be interested in making one. So I've done that and I'll report back when I hear. But otherwise, I was hoping we could try to go a little bit further in the conversation that we started a couple of months or so back about potentially interesting targets. Wendy, do you want to say anything about this first off, about what we'd like to discuss today?
Yeah, so I made 2 slides just to give a small recap which targets we discussed last time, so that makes it a little bit easier to discuss.
Perfect.
So I will share my screen. So last time I spoke that we used 2 strategies to identify new targets for Madurella mycetomatis. On the left, you see a nice 96 well plate. So that stands for the drug screenings we did. And on the right, we did look into Galleria melanella larvae, which, as you all know, forms grains like the mycetoma patients do. And during the process of grain formation, we analyzed the proteome and the transcriptome of both host and pathogens to identify processes important for grain formation. And if we combine these 2 screenings, we ended up with a few pathways which were very interesting. The cytophore biosynthesis pathway with CYT-A and CYT-C highly upregulated during grain formation. Those are an ornithine monooxygenase and a non-ribosomal peptide synthase. The paper which publishes these papers is going to be published tomorrow in Nature Communications. So for those interested in all of the details of the findings, I would refer to that paper. Then in the kinase paper, which is currently under review with PLOS NTD, we stumbled upon the cell wall integrity pathway with, again, 2 kinases in this case, which were highly expressed during grain formation and were also fungal specific. So those would be interesting targets. And then based on the drug screenings, we found the NF-kappa beta potential, the NF-kappa beta inhibitors, which we just talked about on the new series. But we also found a DERK kinase, POM1, which is also a kinase, which might be an interesting target. And at the moment, we're looking at proteins which are expressed under stress. And one of those which we found was the isosome-related protein MMYCO1, and then this number, and that's a potential lipase. So based on the findings we did with the larvae, these are most likely the most interesting targets so far. But of course, others could think differently. But this is at least the main messages of these manuscripts.
Last time we were talking about what was it, a YAC-A kinase, something to do with grain formation? Is that related to one of these, or is that, I forget
Yes, that's related to the POM1. So
Right.
the YAC one was found in Aspergillus, but the POM1 is also found in Madurella.
Okay, great.
Yeah, and
So
there were
that's
like
the
some
Burrell kinase.
tricyclic inhibitors of it that we looked at. There was this compound that was found in a plant called Cujamanin A or something like that.
So these are all, it's possible to make all of these recombinantly? Have they been made, or would they all be new?
Well, for Madurella, there are only a few proteins which I've made recombinant in the past. That was fructose bisphosphate aldolase, pyruvate kinase, and the translationally controlled tumor protein. So those are the proteins which are there. I found one enzyme also recombinantly made by another group, which was looking for enzymes in a process of bleaching. And as far as I know, those are the only already recombinant proteins there. So any protein we would be interested in, we need to either express ourselves or ask somebody to express them. But all the amino acid sequences are there.
Right.
So that would also mean that you could also simply synthesize the plasmid and let it be made commercially by ourselves or by a collaborator. And if they really would like to start from RNA, we do have RNA here as well. So we could also start from RNA.
Okay. Are any of them in any way difficult, like being membrane proteins or membrane-associated proteins?
I don't think so. At least the last one, I know for sure that it's not membrane associated, because the others in that pathway were. I don't think the siderophores are membrane associated. Sean, maybe you can shed a light on that?
They're not, as far as I know. SID-A has been expressed recombinantly previously from Aspergillus fumigatus. And I would say it would be relatively straightforward to do that commercially or in a lab. The SID-C enzyme, that's actually a very big protein. That could be difficult to express just by virtue of its size. And what people do with these NRP synthetases is they generally express domains or fragments of the enzyme. But SID-A, no problem. SID-C, very large protein.
Right. All right. This is great. So this is the I guess, Wendy, this is your preferred shortlist, in other words.
Yeah, this is based on the findings we did in the larvae. There's also another pathway, the zincophore pathway. I didn't state it here yet because the evidence was a little bit less strong. But yeah, and at least from these, I also know that they are relevant in other fungal species. So like in Aspergillus, Sean works mainly with Aspergillus. So if SID-A is already found in expressed from Aspergillus, that's another thing we could do, that we could screen it against two different pathogens. Because in the end, our final goal is to find a molecule and optimize a molecule which is not only active against Madurella mycetomatis, but also against other fungi, so that it can become, in the long run, commercially more attractive. And not per se commercially in that we earn money, but at least that the company will be interested to make it.
Yeah. Okay, this is great. I mean, I guess the way to approach this, as I think we've talked about before, is that in an ideal world, we would be able to access a purified sample of one, either through our own efforts or efforts of the community or some other means, and use that as a case study for doing some screening, so that we can show that the approach works, which of course it will, and show that we can get a tractable hit that we validated, and then use those results to try and generate some funding for the other targets. Question still remains of, how to get a sample of one of these most quickly or most simply. If something has been made before, like SID-A and Aspergillus, then maybe that's a reason for going after that rather than something else, just because it's maybe more likely to work. But any thoughts about either if one is simpler than the others, or any source of protein that we may have overlooked, like someone who could make one of these?
Well, we can make it ourselves. That's not a problem. The only thing which I also thought is, if we only go for one of the pathways, for instance, the cytophore or the cell wall integrity pathway, maybe it's more safe to go at least for 2 different pathways in case one pathway doesn't render the results we would like to. And then hearing from Sean, maybe it's better to go after SID-A than SID-C in the case of the cytophores. And maybe we go for one of the kinases from the cell wall integrity pathway. And if those appear to be very large, then the DIRC kinase.
Yeah.
I think it's not that difficult for us to find out if any of those were ever expressed in Aspergillus.
Right. Okay. The screening that I mentioned, that's possible through what the SGC is trying to set up, will become our capacity to do that will become clearer in about 5 weeks when we hear about the outcome of the grant application we put in. And if we get that, then we'll have more resource to deploy on things like this. So I'll know soon enough whether that's going to be something we can easily do. If not, it's more difficult to do, but if we get it, it's easier to do. So that would be a way of finding small molecules that would at least bind some of these proteins.
Yeah, I could also, one of my technicians could help with setting up one of the expressions, of course. Hearing from Sean that SID-A was done before, maybe that's an easy one to go for then.
Yeah.
And the histidine tag could stay attached to the protein, isn't it?
Well, for the screening that we have in mind from the SGC, then yes, you need the his tag.
Yeah, we use the his tag ourselves as well to purify the molecule.
Right. Okay, sounds very good. I can have a conversation with some people here about which one of those looks like might be the most straightforward to make recombinantly to, but it's not my expertise, but I know people who can comment on that too, just to see what they think, based on the AlphaFold structure. Okay, great. Any other comments about this? Because that's a very, very useful list.
If I can give any assistance with SID-A expression, of course I will. I'm slow to say I'll do it myself because I'm under a bit of financial restriction here at the moment. But if it was a matter of getting the gene made, that wouldn't be a problem. But it's just hands to do the expression. I just don't have that at the moment. Hopefully, it will change in the near future. But if I can give any technical assistance or intellectual input, no problem.
Thank you.
Sean, if we do find a molecule in the long run, would you be able to screen it against Aspergillus as well?
Oh, of course, yeah. No, that will be no problem. Yeah, fine.
Because
And that
then,
obviously, no, I was going to say, obviously, any screening against these SID-A or SID-C would really only work under iron limiting conditions, because they generally are not expressed when iron is available.
yeah, you mean for the in vitro susceptibility later on?
Yes, yeah.
Because for the protein itself, it will not matter, but for
Or to see if it inhibits the fungal growth.
yeah, that's true. For downstream, that makes it a little bit more complicated, maybe. But
Well, I mean,
maybe there's also some insight from the others listening in. So maybe from Bernard, Erica, Louisa, Anthony. I don't have a special preference for any pathway, but maybe someone has.
I know that there was a little bit of similarity between the tricyclic compounds we did work on, well, what was the phenothiazines, but is now the Curaxin derivatives, and that kinase, that DYRK kinase. So I think it would be just a matter of ordering that inhibitor that we found, the Bromley group found, I believe.
Yeah, I mean, have any of these been the subject of hit finding experiments in other fungi? That's just related to what Anthony was saying. I mean, are there known binders of homologous enzymes?
Well, wasn't there I think his name was Dr. Bromley, who did the ethoxycarbonyl beta-carboline tricyclic compound that inhibit DYRK. So I guess it would be a matter of just making that or buying it and seeing what happens. As for Series 7 too, the benzamides, salicylanilides, MQL has a lot of them for very cheap as well. So I think it's just a matter of figuring out where to get the funds to buy certain compounds.
Yeah, I mean, if we're going after this as a target-based approach, then you'd want to make the protein and then be able to assess the binding to the protein. And we can definitely do that here if we have sample of protein and samples of molecules. I think the idea is to make sure we get a binder, and then we can worry about whether or not it's getting in. That would be the approach to take. So yeah, the assay then becomes something which is more biophysics based, or perhaps even one day enzymatic biochemical assay to see if we're inhibiting something. But initially, you'd want to see if you can just find a binder.
I just did a quick look on PubMed for SID-A. There's only one publication where they used SID-A to find an inhibitor, and they screened it against 2,300 compounds, and they found celastrol as a non-competitive inhibitor of SID-A. But that was a quick search. I didn't read it. I just read the abstract.
Yeah, that looks like a possible pan-assay interference compound. It's got this reactive quinone with a C, right? Celestrol.
Yeah.
Yeah, it looks like it reacts with basically everything.
Okay, interesting.
For the cell wall integrity pathway, I didn't get hits. But that was just a very quick search during the meeting now.
I can go back to the SSG CID folks with that with some of those suggestions and see if they are willing to make samples. I think they make small amounts of samples for crystallography. So I don't know if they'd be willing to make and ship samples of proteins elsewhere, but I can ask and engage them in that dialogue. It's certainly relevant to them because it's infectious diseases and it's open, which is their two criteria.
Yeah, and depends on how they normally make them. But if they make them in E. coli, they can also simply ship the E. coli.
Mm-hmm.
So that we induce the expression of self.
Right, right. Okay, that's great. Thank you very much. No further comments on that list. I guess it's an action on all of us to consider what has been done on similar proteins previously and any brainwaves about how we might access samples of proteins, other than doing it ourselves, how we might access samples of proteins with help from others. All right. Thanks, Wendy. Thanks, Sean. Okay, so I think the only other thing that we were going to do today was then talk about the series 2 paper. That's right. So Clementine, did you want to say something about where that's up to?
Yes. So I have a draft basically made, but rather than, share a multi-page document, I thought I would just do a quick presentation if that's all right. Let me find, here we go. So I'm assuming you guys can see my screen.
Yep.
Can you see the right screen or the presenter mode one?
You're in the wrong mode.
Yeah, hang on a second. Okay, there we go, I think.
Yep.
So, I mean, a lot of these are questions that will take a little while to discuss, but basically, this is our compound, and because I've started using this terminology fairly recently, but I'm calling this the C2 aryl ring, and this one the C4 because that's what other papers have done. So that's what I've been using in this paper. So we've screened lots of compounds, and we haven't actually finalized the results of the efficacy or the toxicity and efficacy testing or any of the in vivo stuff. So one thing that I would like to ask about is what timeline we might be working on this year in terms of the larvae. But the results that we do have so far, we have looked extensively at this C2 ring, different substitutions. The extremely obvious trend is that if you have anything with like a phenyl instead of a pyridine, it doesn't work. We have a few results that we've only just recently made, so we've recently made the compounds, so we don't have the in vitro screening for them yet. But once we have those, we'll have a pretty, comprehensive set of different substitutions and so forth. There's obviously not that much that you can get from this information, but there's a little bit of a trend with the positioning of electron withdrawing groups versus electron donating groups. On the other side of the molecule, the C4 ring, we have the same thing where if you have the phenyl group, it doesn't really do anything, but we haven't made many variations with the pyridine ring. So again, there's a couple that we don't have the results for yet. I don't actually know what happened to this molecule. I think it was made by the Uni of York, and Peter isn't here today because I'd like to ask where that went. I can't seem to find the results for it. But again, hard to draw a whole lot of helpful information here. With the in vivo screening, this is compounds. A lot of these are compounds that I tested and the results weren't entirely usable. I'd like to ask what do we want to include, like what's worth including from these compounds, and which ones are we likely to end up having results from? So that will obviously impact what we're putting in. And I know that the biologists obviously put the survival curves in usually. I've just put this into a table because that's more how my brain works with it. If people have comments on how to display that data, I'd like to hear them, but I was trying to, I guess, kind of squish everything relevant to the in vivo screening into one table. In terms of the discussion, again, this is a fairly straightforward paper. There is a little bit of stuff that I found about, so this is our lead compound, and other people, in particular this group, Pieroni's group in Italy, they have been making really similar compounds for tuberculosis, and they have theorized that their 2-aminothiazoles are similar in structure to phenothiazines, and they know that phenothiazines can block these enzymes, so that's their explanation for why they work in tuberculosis. That's mostly the only leads that I have for explaining the mechanism of action of this. The other lead that I was hoping would go somewhere but didn't is that this compound here is both of these compounds are calcium channel blockers, but obviously this one is nice and potent against mycetoma, and this one doesn't seem to be, although those results are still being finalized. But basically, it hasn't been doing too well in high-low screening, so alas. I think, yes. I put a few of these things on GitHub before, but we'd like to have those log D calculations if that's possible, because I know you guys have looked at that before, the frenaramols, and I think also with the catoxines. I'm not 100%. We've talked obviously about knockout models, etc., and molecular modeling, because a lot of the known drugs, specifically the azoles, we know target the CYP51 enzyme. It would be good to know if these target the CYP51 enzyme, because that would, give us a lot of information. But we don't have a knockout model at the moment. I'm not aware of anyone who has the capacity / skill set to do molecular modeling. And from what Wendy was saying on GitHub, I'm not sure how useful it is to do those synergistic co-administration studies, because your results are kind of only useful if they are positive, from what I gather. I mean, obviously, negative data is useful in its way, but not as useful as it could be. There are a few compounds that, we have made lots of compounds that looked at other things, but we don't have kind of complete enough sets of any of them yet. So I have a couple of those here. We've looked at different, isosteres on this C2 side. If we get the results for these ones, I'll probably include that. But if not, I don't know that it's that worth mentioning. The same with the methylation of these compounds, where we were trying to look at methylating the amine, and so far haven't actually been able to make and purify any of these compounds, unfortunately. And then the Uni of York started looking at some alternative linkers, and they did make some, but they're not kind of direct, mostly they're not direct analogs of our lead compound. So it's a little bit difficult, but also they didn't work very well. So I think the Uni of York was not going to continue with that unless we had a really strong reason for them to. So yes, just to try and sum all of that up. What else are we going to make? Are we going to make any more compounds with this? I know, obviously, Peter with the Uni of York and us with UCID, we would like to decide fairly soon so that we can organize things for the next semester. Do we want to continue making these aminothiazoles at all? Do we think there is much use to making any more of them? And yeah, what is the timeline for the in vivo, in vitro testing generally?
That's
All right.
great. Thank you. That's really good. Can I jump in with a quick question where you were talking about the channel blocking, the calcium channel blocking?
Yes.
You mentioned that -176 is a calcium channel. How do we know that?
I kind of assumed that you had put that on the README for the series. There's basically a paper somewhere where they've identified it. And I'm not actually sure where this one is identified as being a calcium channel blocker. I can't remember off the top of my head. But I don't know enough about the relative IC50s or MICs here, in that this is a far more potent inhibitor of that channel than this one is. So perhaps, if we do end up finding that this has some activity but not much, maybe that is a clue, but I don't know if that's a bit grasping at straws.
Is there, I mean, I guess the question for Wendy is what is there a way of seeing whether or not this mechanism is operating for Madurella, mycetomatis?
Yeah, the problem is we can't make knockouts yet, so that's the strategy which you would normally use, but we can't. Yeah, I need to look in literature, maybe if there's a certain enzyme assay you could use. I don't know. Yeah. Clementine, you asked about the timeline. How many compounds do we still need to test?
So
Dunya said that the list is smaller than yours, so...
Sorry, say that again?
Dunya's list is smaller than yours, so...
Yes, I mean, that's, I think, because some of the ones that I was looking at last year, because, we started with the in vivo testing before we'd finalized the MIC results, the cutoff, includes too many. But basically, I think we would like to publish this paper soon. So whatever is the most feasible number of compounds that would give us some information, it doesn't have to be every single one that had an MIC of less than 2,
No, but there you can help us with which ones are most important for your comparison. The MIC is one thing, but the comparison you want to make is the other thing, of course.
Yes, I can certainly put together a list. It's a bit hard to do instantly, but I could put together maybe like a half a dozen, 4, something like that are the most important.
So if you can prioritize, these ones are essential, these ones great if they could be done, and these one, please do them only if you have time. If you
and a million larvae, yep.
Yeah. That would help us with planning as well, because for Dunya it's the first year, of course.
Yes.
That she is going to do this, so it takes her much more time than it would have done with Ma, who has done these for 4 years in a row.
Yeah, I mean, I think ultimately, although I would like it to be otherwise, I don't have extremely high hopes for their activity. And that was, I didn't actually exactly say this question, but with the phenaramols and the chitoxins, which are the most kind of, complete 2 series so far, were there many compounds that did have in vivo, like good in vivo activity?
Well, with the phenaramols, I think it was 5 or 6 on 30.
Okay.
Yeah.
Because, I mean, at the moment, it feels like an extremely bad ratio that, we've tested 100 compounds and found one, like only the lead compound has any in vivo activity. But obviously, we haven't actually done in vivo testing of many of them. So that's,
Yeah, and the problem is that we cannot predict from the in vitro results which one will be very effective in vivo, because it's not a clear-cut correlation between MIC and in vivo efficacy, because it's also the biofilm-like structure, the grain, which interferes with that. It's also how stable is the protein when you put it in serum or in hemolymph in this case. Yeah, so these are all things which we will find out during the in vivo stage at this time.
yes. I can see Louisa has raised her hand.
Yeah.
Sorry.
Yeah.
Yeah, no, thank you. I'm sorry, I just I couldn't speak before. But one question, do you know if the CYP51 from like fungal CYP51 is like closely related to the CYP51 from Cruzi? Because if so, we do have some modeling available at TNDI. I could check if we could do some C51 inhibition enzyme assay for representative compounds for the series. It's something that could interest you, maybe. And also for the log D, I can do the calculation if I have the descriptors of the compounds.
Okay, great. Well, with the log D, I put I have a Excel sheet. If there's anything missing from it, let me know and I'll put it in.
Cool. I'll check it.
Yeah, thank you. With the CYP51, I am not sure how similar it is to other CYP51s.
Yeah, I would assume it is. But I just wanted to see if you have sort of like any one of the MycetOS used that sort of comparison before, because if it's useful, I can probably do it. I don't know if Matt or Wendy has any experience on this.
Well, CYP51, we used Aspergillus model to predict, to build a model for this one in the past. Madurella mycetomatis has one. Madurella fahali has apparently two. But in this case, I don't think CYP51 is the target.
Yeah,
for the February model.
I'm just because you just wanted to see if there's anything related to it, but that's just kind of a counter screen. But so it's only from Clementine slide.
Yeah, Wendy, I was simply trying to think of like
any pathway.
If we could do the modeling and say it
it
doesn't.
Yeah. But I mean, only if it's useful. So it's available. We do have the modeling, and we have also capacity for the enzyme assay if it's useful.
You could always try. I think in the past the phenarimols and itraconazole they also work against that CYP51. So you could try.
So they did work?
Yeah. They were like phenarimols or like inhibitors of CYP51. So maybe we could. What I could do is try to see if I can do some modeling, see where is available at the NDI, and then if we have anything interesting, then I would try to go for the assay. But like again, it's the T-Cruzi CYP51 assay. So we have to see if there's any correlation in there.
Yeah, so what you could do with the modeling is see how well it fits. If there's a complete mismatch, then don't go for the enzyme assay.
Exactly. Yeah. Agree.
Yeah, that would be great.
Okay. Yeah, I'll have to dial off now, but I'll get the list in the GitHub and do the log D and see about the molecular modeling.
Okay, thank you.
Cool.
That's great.
Cool. Thank you all.
Thanks, Louisa.
Bye bye.
So, could I add on this about CYP51? I think we did one of the MSc project students did some modeling last year on aminothiazoles. So she docked the human CYP51 and then created a homology model using the sequence of the Madurella analog. On a first glance that I'm looking at the binding scores, there wasn't much difference between the human and the Madurella mycetomatis one, but also I'm a bit skeptical about the results. So, we could, if you think it's worth it, we could repeat it. Like Luisa maybe could look into this, but it's possible, yes. So you can find it's possible to make a homology model, yeah.
Okay, so have you shared those results before, Erica? I remember you talking about them, but is there like
that
a
was
file
last
somewhere?
year, right? Let me see, I will find, the file and then put it again in. But again, we treat them with caution, right?
Yeah, because I think also some of the compounds that you guys were using for that work
Yeah.
weren't good inhibitors.
No, but we used yours as well, the ones you sent. Remember you sent like C?
Yes, okay.
I think they are in the modeling, yeah.
Okay.
Yeah. I will create a table and see whatever data I find from the student, and I can upload it on the issue with the paper on that
Yeah, that
issue.
would be good.
Okay.
Yeah.
Yeah.
Yeah, I mean, any in silico model, I think it needs to be able to it needs to be able to explain a little bit the rank order of molecules in one chemical series, provided that the observed potencies are significantly different, right? So if you have an order of magnitude difference in potency and the structures are similar, the docking needs to be able to explain that a little bit, not the fine detail, right? And if you change the structure of the molecule, then you may change the ability for the molecule to get into the pathogen or whatever. But if you've got a defined series that is structurally similar, you've got to have some kind of ability to reason that rank order of molecules. If you don't, then it doesn't really help you. It doesn't really help validate the target. The calcium channel, I guess it might be useful to go back to that paper and see how that was established and whether or not we could do something related to that methodology to see if, again, it doesn't really help us in this case, right? But it'd be, I'll go back to that paper and see how they did that because
I can't, let's see.
We would want to know if there is something equivalent in Madurella. If that is some other organism, which I'm assuming it is.
Yes, that's
okay. Great. That's good. Thanks. All right. We're coming up to the end of the hour. So did anybody want to follow up on anything that Clementine just said about Series 2? I mean, from my perspective, I think we need to minimize the number of extra molecules we're making this series because I think that you're right. We need to try and focus in on publication. And unless we come across some amazing compound that does really well in vivo, I think it's reasonable to say that we should put our efforts elsewhere. But we do need to get something publishable out of it, which may require, the additional testing of select molecules to make a better story.
Yeah, I mean, from my perspective, there's not a whole lot of reason to make new molecules, but it would be nice to kind of be agreed on that so that we can figure out something else for our first years to do.
Yeah.
Yeah, I have a few in making, but maybe this month, maybe the next, so maybe in total no more than 10, and then that's it. Oh, and it's the wheat gift, but it's okay, the wheat gift. We can
I mean, we also have 2 or 3 more that have been made, and we'll send them for the sake of completeness. But
the N-methyl analogs that you showed, was I right in thinking that you have successfully made one of those?
So I kind of tried it myself and then got a student to do it, but I haven't been in the lab So it's been a bit hard to kind of keep track. And I
Yeah.
think what the student ended up doing was making the imine, and it needs to be reduced separately.
Right.
And I keep meaning to try that and not finding the time because I'm not in the lab, but I don't see any reason why it wouldn't work if you did a reduction to get there.
Yeah.
There is a chance that we might have somebody, like a short-term, student who could maybe do that. But I mean, I'm not sure how likely that is or not, but that would be like August, September kind of timeline.
Right. I mean, of all the structures, that is that's makes sense to make, right? Because it's a little bit different from the others, rather than another R group. Yeah, that would help.
Yeah.
Yeah, I mean, for the Whitgift samples, I guess we were interested in seeing whether or not we could do that. I forget, Erica. They had done the previous step, but the final step didn't work.
It's actually from step 6, so it's the hydrolysis with the sodium hydroxide that didn't work. So this is what the RNMR showed. So they are repeating that. The same process, they're just repeating and then monitoring the reaction with TLC instead of just stopping it at 30 minutes. I think that was what the protocol said.
Okay.
But they won't start until September.
Yeah.
I think Erica, it would be well worth
Yeah.
seeing if your students can try doing, because they were making it like from memory, it was already alkylated before they did their
Yeah.
Hansch
So this is
synthesis,
yes,
right? That's the goal. Whereas
Well,
we were
I
trying to do the Hansch and then
yeah.
alkylate it. So
Yeah.
maybe they can try that
The-
too.
They will try that too as well. Yes, it's not going to be though in the timeline, or is it urgent? You would like it now?
I
Yeah, if
mean,
you would.
we would like it now, but I'm guessing that
Yeah.
the biologists will also be going on holidays, etc. So,
Yeah.
Yeah. Yes. Okay, we'll give it a try. And we're also doing the amides that I told you and added a few ones with with the pyridine ring. Yeah, purification is a pain.
Yes.
But
Yeah.
yes, but we're trying other reaction with chloride instead of carboxylic acid. I think it works a bit better. So I may have a few of those, which
Okay.
we can compare with the lead that doesn't have the amide. Yeah.
Yes.
Yeah.
So I think we talked last month anyway about maybe having a session separately to figure out what compounds we might do in future, for universities specifically. And given that we're running out of time, that's probably a good idea to leave it until then.
Sure.
But yes, if anyone has thoughts on what might be whether series 7 maybe.
Yeah, I was going to say the
Most students.
benzanolides are about one step from an acyl chloride and an aniline basically being heated together in a solvent.
Yeah. So I think that's probably doable for the Uni of York. I'm not sure if we can get our 18-year-olds to do it. We'll see.
Yeah, there are some nice new peptide coupling reagents we could maybe employ for that.
Yeah, that
Yeah,
would be
there's
good.
EDCL. Bernard used that. Oh, there's a cat.
Yeah. Well, he's upset with me for being working at this time of night.
Up late, yeah. Okay. Good prompt. Thanks. Yeah.
Sorry.
Yeah, no, that's good. Right. We should wrap up. Hopefully we've covered everything. We should have the schools session meet up sometime, I mean, first half of July might make sense, given that people are going to be away, second half of July and August. I'm around for a bit of August, so we could do it then, but maybe we should aim for sometime in July. Maybe we can do a little get together on one of the issues and try and figure out a time to do that when we can make sure everyone's there.
Yeah, I'm happy to try and get that sorted.
Thank you. Great. Fantastic. Okay. All right. Anything else anyone wants to raise that we haven't, just super quick, that we haven't talked about yet? Otherwise, obviously dump things on the issue that we've had from today. Okay. If not, thanks very much, everybody. And we will see each other. I can't remember if we
Bye,
have a
everyone.
meeting. We don't, right?
I think we're skipping July.
We're skipping July.
I think.
Okay. All right. All right. So we'll see each other soon. And any updates, put them on the GitHub issues.
Yeah. Okay. Bye.
Thanks all.
Bye.
Bye.
Bye-bye.